Hi everyone, welcome or welcome back to my channel. This is Sagnick Dutta. I'm a second professional MBBS student at Burdwan Medical College and Hospital, West Bengal. This is the second video of this series. In the previous video, we have talked about erythrocytes, that is the red blood cells, and we have also talked about the process of formation of RBCs in the body, that is erythropoiesis. We have also talked about hemoglobin, glycated hemoglobin, and the metabolism of hemoglobin as well. So in this video, we will talk about some investigations and experiments related to RBCs, and we will also talk about blood indices, which actually help us to compare between the blood of two different persons and also help us determine the pathological conditions in a person if present. So without much ado, let's get into the computer screen and start learning. Now let us start with some investigations related to RBCs. The first and foremost experimental investigation that is done is the erythrocyte sedimentation rate, which is also known as the ESR. Now what actually is ESR? ESR is the rate at which the RBCs settle down when a blood sample is placed in a vertical tube. Now what are these tubes? The tubes used are basically the Wintrob's tube or the Westergren's pipette. Now, what is the process behind calculating this erythrocyte sedimentation rate? What is done is we take a test tube or these tubes, the Wintrob's tube or the Westergren's tube, and a blood sample is taken and it is allowed to settle down for about 1 hour, and what is seen is that the plasma separates above these red blood cells. The height of the column of these supernated plasma is basically the ESR. Now, talking about the normal values of the ESR, it is 0 to 5 millimeter at 1 hour for males if the Wintrob's tube is used, and is around 0 to 8 millimeter at the end of 1 hour for females if the Wintrob's tube is used. Now when we have known about the normal values, let us study the physiology behind the ESR. Basically, when we talk about our RBC, it has got some negative charges around it. As a result, a negative potential is created around it, which is known as the zeta potential. And because of the presence of this potential, what happens is the RBCs repel each other and they do not clump together. But when the blood is made to stand still outside the body, the RBCs form an aggregate and pile on each other like a stack of coin. And this stack of coin is basically known as the rouleau. So when the blood is made to stand outside the body inside these tubes, the Wintrob's tube or the Westergren's pipette, rouleau formation begins. Due to the presence of some asymmetric macromolecules in the plasma, the rouleau formation hastens when outside of the body and the RBCs settle down. These macromolecules are, for example, fibrinogen, gamma globulins, which are present in the plasma, and if these are present, the ESR actually increases. The process of rouleau formation increases. So the conditions in which such proteins are increased in the plasma will therefore exhibit an increase in the ESR. And these proteins increase in conditions like tuberculosis, malignancy, anemia, rheumatoid arthritis, and so on. Now, a very important thing that we need to keep in mind is ESR do not have any diagnostic significance. What I mean by it not having any diagnostic significance is that ESR increment or decrement doesn't symbolize a specific pathological condition. As we have seen earlier that ESR increases in case of tuberculosis, it increases in case of malignancy, it also increases in case of anemia. So if an increased ESR is found, we won't be sure which pathological condition is present. Thus, ESR is not of any diagnostic significance. But what actually ESR does is it provides a prognostic significance. Now, prognosis means the progress of a disease. Once a disease is diagnosed, the ESR of the patient is recorded time to time, and then with continued treatment, ESR is measured at regular intervals. If the ESR decreases, it means that the patient's condition is improving. Thus, ESR do not have any diagnostic significance, but it does have a prognostic significance, and that is why measurement of ESR is important. Now, the second investigation done is the packed cell volume or the PCV. Now, what is the packed cell volume? It is actually expressed as the percentage of RBCs present in the unit quantity of blood. This PCV value is also known as the hematocrit. In any unit quantity of blood, roughly 45% volume is of the blood cells, 55% is of the plasma. Now, out of these 45% cells, most of them are RBCs. Thus, we can say that 45% of the blood is about RBCs. So, the normal PCV values are, for males, it ranges from around 40 to 45%, and for females, it ranges from around 38 to 42%. Now, how do we actually determine the PCV? We do it by the Winthrop's method. The Winthrop's tube is filled with the blood sample. This tube is then kept in a centrifuge and is rotated at around 3000 RPM for 30 minutes. This creates an optimum centrifugal force. The RBCs are packed together at the bottom half and the plasma separated, positioned at the top half of the blood sample. Basically, 4 layers are created in the tube from below upwards. The first and the bottommost layer is of the packed RBCs. After the layer of RBCs is present a buffy coat layer of the WBCs. Then above it is present a thin dark line of hemolyzed RBCs above the WBCs. And at the topmost layer is present a layer of supernatant plasma. So the layers are basically, the topmost is of the plasma, next is of the lysed RBCs. The third one is of the WBCs, and the last one is of the packed RBCs. Right. Now, what is the significance of PCV? It is also forming a layer. The ESR was also forming a layer between the plasma and RBC. But what is the actual difference between these two is we get four different layers in case of PCV, which is not obtained in case of ESR, and that is why PCV becomes more clinically significant. The first significance is of anemia. Since the RBC count is decreased in anemia, the PCV will be lower. The PCV will be less than 38%. So if a person's PCV is found to be less than 38%, he or she may be termed as anemic. In case of polycythemia, increased RBC count is seen. As a result, PCV will also increase and its percentage will be more than 48%. Now, in cases of conditions where there is loss of plasma, that is, there is hemoconcentration. For example, in case of diarrhea or vomiting, the plasma concentration decreases. As a result, PCV will increase. Let me explain this a little clearly. Look, PCV is basically a relative proportion of the cells and plasma. If plasma decreases in a unit quantity of blood, the cells will relatively increase in proportion. As a result, PCV will increase. Now, in case of jaundice, normal plasma is straw colored, right? So, if we see the tube while measuring the PCV, the plasma will be of straw color. But in case of jaundice, the plasma bilirubin increases, and bilirubin is a yellow colored pigment. It will then turn the buffy colored plasma into a yellow colored plasma. Thus, if PCV is measured in case of a jaundice patient, the plasma won't be straw colored, rather it will be yellow colored. Now, let us talk about the blood indices. What actually is an index? Index are basically numbers or values which are related to the RBCs, which are derived from some observations and calculation. The first blood index which is used is the PCV, which we have discussed a little few minutes ago. Now, the second important blood index is the mean corpuscular volume, that is the MCV. Now, from the name itself, we get to understand that mean is the average, corpuscle resembles the RBC, and volume. So, it is basically the volume of a single RBC. Now, how is it measured? A blood sample is collected, the RBC count is done, and the PCV is measured. If the blood sample has a certain number of RBCs and all those RBCs when packed together have a certain PCV, we can calculate the average volume of one single RBC in the formula. For example, if X number of RBCs occupy Y volume of PCV, then one RBC will have a volume of Y by X. Right, that is the mean corpuscular volume. The normal MCV range extends from 78 to 93 cubic micron. If MCV is greater than 93 cubic micron, then we will call that RBC a macrocyte, which is usually seen in case of megaloblastic anemia. Right. Now, if the MCV is less than 78 microns, cubic microns, then we will call it a microcyte. And this is very much prevalent in case of iron deficiency anemia. And that is very prevalent, right? If there is iron deficiency, the formation and growth of the blood cells is hampered. As a result, they decrease in the size, forming microcytes. The third blood index is known as the mean corpuscular hemoglobin, or the MCH. This is the average amount of hemoglobin which is found in one single RBC. Now, if a blood sample, hemoglobin estimation and RBC count is done, if a certain number of RBCs contain a certain amount of hemoglobin, then one RBC will contain a certain amount of hemoglobin, and that hemoglobin concentration is known as the mean corpuscular hemoglobin. Thus, we can write mean corpuscular hemoglobin is equal to total hemoglobin concentration by the total number of RBC, or the RBC count. The normal MCH range is from 28 to 32 picogram. This MCH is reduced in case of microcytic anemia. When the cell itself is small, how will it contain more hemoglobin? Thus, the hemoglobin concentration also decreases in case of microcytic anemia. Now, the fourth blood index that we need to talk about is the mean corpuscular hemoglobin concentration, that is MCHC. This is the relative percentage of hemoglobin in a single RBC. The normal value is from 32 to 38%. Mean corpuscular hemoglobin is normal in case of megaloblastic anemia, but MCHC is reduced. This occurs in case of megaloblastic anemia. Look, in case of megaloblastic anemia, what happens is the cell size increases, but the hemoglobin concentration remains the same. For example, in 1 unit blood, there was X amount of hemoglobin. In case of megaloblastic anemia, what happens is the blood cell sizes increase, but the quantity of hemoglobin remains the same. As a result, the mean corpuscular hemoglobin is normal. But because the size of the blood cell has increased, the relative concentration of hemoglobin inside the cell decreases. Thus, mean corpuscular hemoglobin concentration decreases. Now, talking about the fifth and the final blood index is the color index. Color index is the ratio between the hemoglobin percentage by the RBC percentage. Look, hemoglobin level, if is around 15 gram percent, it is considered to be 100% hemoglobin. So, if a patient has around 10 gram percent of hemoglobin, it will be around 66% hemoglobin. RBC count in million per cubic millimeter is converted to percentage values. That is 5 million per cubic millimeter is considered to be 100%. So if a patient's RBC count is around 4 million per cubic millimeter, it is considered to be 80%. And then their ratio is found out, and that determines the color index. The normal value of the color index ranges from 0.8 to 1.1. Now, if a patient has color index less than 0.8, he or she is said to be hypochromic. If in the normal range, then known as normochromic. If is more than 1.1 is known as hyperchromic. Now, hyperchromic condition is not seen usually because you imagine the concentration of hemoglobin doesn't increase in any case. Thus, hyperchromic condition is not seen. But these two conditions can be seen. I hope you liked the video and it was helpful for you. If you did like the video, hit the like button, subscribe to the channel and share it with your friends and to whomsoever it may concern and be helpful. Thank you.